Welcome to the next set of videos on the principles of immunotherapy. It's a three-part segment because it is quite complex. And the first part will be looking at the immune system and how it normally functions. By the end of this presentation, I hope you'll share my sentiment that this is a very complex system and fascinating. And it, at the same time, is very important to understand in order to understand how immunotherapies work for treating cancer. To start, we have to look at the lymphatic system. The lymphatic system is a series of channels, lymph channels, that drain our tissues, and they drain them to regional lymph nodes, shown here in green dots, throughout our body. And as you'll see as we go through the talk, this is where the central process of the immune response to various things like exposure to infectious agents like viruses or bacteria and cancer. Other important components of the immune system are the bone marrow and the thymus gland, which we'll get to in the next couple of slides. But first, the bone marrow. This is where the immune system starts. In the bone marrow, we have hematopoietic stem cells, and these stem cells are undifferentiated. They differentiate, when appropriate, to three main component cells: red blood cells, white blood cells, and platelets. And these are differentiated cells. We went through differentiation when we talked about pathology under the microscope of cancer cells, in that the stomach cells are differentiated cells, and the undifferentiation of those cancer cells that we see can be graded as well, moderate, or poorly differentiated. So they're undifferentiated. Incidentally, similar to stomach cancer, cancers of the blood, or leukemias or lymphomas, are undifferentiated and look more like hematopoietic stem cells compared to their differentiated component. Now, this is a simplistic way to look at it. It's a little bit more complicated, where we actually have the hematopoietic stem cell, which is called multipotent, which can form two main types of subset stem cells: myeloid and lymphatic. The myeloid stem cells can differentiate further into different components: erythroblasts that become red blood cells, megakaryoblast, which become platelets, myeloblasts, which become basophils, eosinophils, or neutrophils, and monoblasts, which become monocytes. And then the lymphoblasts, which can become different types of lymphocytes, B cells and T cells. The white blood cell then, which encompasses all of these types of cells, can be either granular, which has little granules in them that are their effector function, how they kill their target, or agranular leukocytes like B and T cells. Red blood cells, of course, transport oxygen to our tissues so that they can function. Platelets are involved in clotting. We talked about how blood clots are common in cancer, and that platelets are how those blood clots form. And then the remainder of this talk will be focusing on the white blood cell component, which is our immune system. You should see that these different cells last for a finite amount of time. They turn over routinely. Red blood cells on the order of 3 or 4 months, platelets on the order of a week, and white blood cells can be around for anywhere between days to a few months. And so they're turning over. These cells are continually dividing as appropriate and differentiating into their components as appropriate. And when we get to the toxicities of our therapies, particularly chemotherapies, where we learn that chemotherapy inhibits dividing cells, there is collateral damage on other cells that are dividing, like our blood cells that are repeatedly dividing in the bone marrow in order to replenish our counts. And this is why we get low blood counts to begin with. So now that we've learned about the cells within the immune system and how they get there, the other thing to note is that, remember, each of these cell types have the same DNA inside of them. It's just that they're expressing different genes of our whole gene portfolio in humans to make them their specific cell. This figure is flipped on its side compared to the previous slide, where the progenitor cells are at the bottom and they're differentiating upwards. So the next thing on this slide that we learn is that there are two components of the immune system, two main arms. The myeloid progenitors make the innate immune system, and the lymphoid progenitors make the adaptive immune system. The innate immune system is a general response. It's not specific, but it's our first line of defense against pathogens like viruses and bacteria and cancer. And because it's general and nonspecific, its benefit is that it's a quick response. It's the first defense to exposure of these external or endogenous internal problems. There are a number of cells that mediate this immune response that is called the innate response. On the other side, we have the adaptive immune system, which is an acquired immunity. It's learned based on the specifics of the given pathogen or cancer, and therefore it's slower. It takes time to learn that, but it's much more specific. In addition to that, after clearing the infection or the cancer, there are memory cells that linger, so that if that exposure recurs, like a repeat infection, then your response is faster because you have those specific memory cells to that specific problem. The two main effectors of this acquired immunity arm are T cells and B cells. Other topics that are important to understand that we'll go through in the presentation are antigens and epitopes, antigen presenting cells, APCs, major histocompatibility complex, or MHC, also known as human leukocyte antigen, or HLA, and cytokines. So we're going to go through each of these important cells and concepts in turn, and starting with antigens and epitopes. So what is an antigen and epitope? In order to understand that, just a quick refresher on the central dogma of biology, that DNA is the blueprint of all cells, that is a sequence of base pairs, and that DNA can be turned on, a gene can be turned on through transcription to RNA, messenger RNA, and the mRNA is translated into protein through the specific sequence of bases that dictate which amino acid will go in which sequence. And the importance of this is that this dictates the function of the protein. That's shown here, that the specific sequence of amino acids as dictated by the DNA then will fold on itself, because each of these amino acids has a three-dimensional structure that helps it fold on itself into a three-dimensional structure, and sometimes even bind to each other with different gene products, different proteins coming together to make mega proteins. And we're going to see that when we go through various proteins important in the immune system. Something of importance is to understand that the length of a protein ranges depending on which gene and protein we're talking about, from very few amino acids in the hundreds to even thousands of amino acids. And so the majority of which you can see here are in the 100 to 800 range. Proteins fit together like a lock and key. And so the three-dimensional folding is important, again, as dictated by the sequence of amino acids, and they can come together and bind to each other. Similarly, when we were talking about targeted therapies, we looked at how proteins can bind to each other like ligands and receptors in a lock and key function. And this binding can then activate or lead to conformational changes in the three-dimensional structure that then lead to activation of proteins that then can bind other proteins and activate them. And this protein would then be changed in its three-dimensional structure to be able to bind another protein, etc., down a signaling cascade where proteins can bind to each other when they're activated and cause this protein-protein interaction that is specific to the shape that they are, which is specific to the sequence that the amino acids are in. And now we're going to see how the immune system has protein-protein interactions, where an antibody, which we had talked about in targeted therapies, has a specific part of it called the paratope part that can bind to specific proteins at a specific site called an epitope. And so an antigen is a large protein or piece of protein, like called a peptide, that has a part that can be bound in a lock and key manner that will allow for it to bind. Notably, this same antibody would not be able to bind to other parts of that same antigen. It's very specific and unique to this one region. That is, again, specific to the amino acids in that area. The next thing to understand is that antibodies can bind to a protein or an antigen in a sequence where it happens to be a linear part of the protein, so that all the amino acids are consecutive in the protein, a continuous linear epitope. In contrast, it can also bind to the three-dimensional protein that when you unravel it, called denaturing it, the actual amino acids that it binds to are not contiguous. They're discontinuous. They're in different regions of the actual linear protein, which is, again, speaking to the importance of the folding of these proteins. B cells make antibodies, which we're going to learn, and they can bind to either type of epitope, a continuous epitope or a discontinuous epitope. Whereas T cells, as we're going to see, are really only able to bind to linear epitopes. Okay, so now that we understand antigens and epitopes and how this protein-protein interaction is like a lock and key, let's dive into the different cells. So let's start with the innate immunity and start with those cells called neutrophils, monocytes, which can then differentiate into either macrophages or dendritic cells. The first concept is phagocytosis in this innate immune response. And cells like macrophages or neutrophils can recognize specific products of bacteria or other pathogens that are called pathogen-associated molecular patterns. They're not specific to a given bacteria. They're just generally expressed across various pathogens. And these PAMPs are recognized through protein-protein interaction by receptors on the macrophage and neutrophil called pattern recognition receptors, of which there are several types. One common one is a toll-like receptor, of which there are multiple subsets by various numbers you can see here. And so again, it's not a specific response, but it's a generalized response as a first line of defense in the case that we are exposed to things like bacteria, that we can quickly recognize these PAMPs, internalize them, and neutralize them. So this is called phagocytosis, when these bacteria are getting internalized and killed. Now, in addition to these PAMPs response to external or foreign microbes like bacteria, we also have damage-associated molecular patterns that are released by our own cells, called host cells. And in the event that there's a cancer, for example, that's proliferating and having a lot of cell death and turnover, it is releasing DAMPs. And another mechanism then that macrophages have is that they can, through the same pattern recognition receptor, recognize cancer cell proteins and DAMPs and internalize cancer cells and neutralize them. In addition to that function of phagocytosis and neutralizing the cancer cell internally, they can also, macrophages can also digest these proteins from the cancer cell and express them on their surface. And we're going to go into that in great detail. And this is the first introduction then of an antigen presenting cell, which can take components of the cancer cell and express them on the surface on a specific molecule called the MHC Class 2 that we're going to talk about. And that can help to train other immune cells, namely T cells, and elicit and initiate the adaptive or learned arm of the immune system that's teaching our cells about the uniqueness of these cancer proteins. So this is now the introduction of the cancer immunity cycle, where we start with tumor cells in the place where they started or in metastatic sites. And as they turn over and die, they release those DAMPs, damage-associated particles, and they can travel through the lymph system to lymph nodes, where in the lymph nodes there are an important cell called dendritic cells. And these dendritic cells can uptake these DAMPs, as I just showed you on those receptors, and become activated, digest those proteins into smaller pieces, and then express them on the surface through MHC receptors that we're going to talk to in detail in a moment. And there are different kinds of MHC receptors that will then train T cells in the lymph node, that then this trained army can proliferate and become more T cells that recognize that specific antigen epitope, and then travel back through the bloodstream to areas where there's tumor cells and recognize the tumor cell that's expressing those very antigens and kill them. So this cycle is really what we'll be focusing on a lot in this segment of immunotherapy, because it relies on tumor cells shedding antigens, our immune system uptaking them and expressing them on the external surface on antigen presenting cells like dendritic cells. And that will help to train our army of T cells to go and find the cancer cells and kill them. So a little bit more then on how this is a specific response. We go back to the genetic changes that we talked about, single base pair changes in the DNA, or small insertions or deletions that leads to a frame shift and a change in the triplet codons downstream, and or fusions that ultimately will lead to different amino acids in the protein, which is what the definition of cancer is. And this different amino acid that is replacing what should be there is called a neoantigen. It's a new type of antigen that is non-self and that our immune system will recognize as being abnormal, hopefully. And so the normal version of the protein without the mutation or change versus the novel oncoprotein would get uptaked by antigen presenting cells like that dendritic cell we just talked about inside of a lymph node, and through a processing system called a proteasome that degrades these proteins into smaller pieces or peptides, where if it was a normal peptide, the normal protein would get upregulated on and expressed on the MHC molecule. If your T cell bound there, it would not activate. On the other hand, with an abnormal sequence of amino acids here, and it's being expressed on the MHC molecule, then a T cell, as we will see, that is specific to this lock and key could be activated and proliferate. So T cells would recognize non-self versus self. And we're going to talk a little bit more about that when we get into T cells, how specifically that happens. So for the first part of the innate immunity, we talked about the phagocytes, macrophages, neutrophils, and how they are the first line of defense to various things, whether it's an external pathogen or internal products from cancer cells, as well as dendritic cells and other antigen presenting cells that are the liaison, the bridge to the innate, from the innate immunity arm to the adaptive or acquired immunity arm, to teach or to select for specific T cells and B cells that we're going to learn about, that will then be much more specific based on the specific characteristics of the cancer cell. And just to recall and remember that the innate response is therefore not specific and a very quick response based on these general properties and general receptors that are expressed, pattern recognition receptors. And then over time, the adaptive response that is much more specific, but it's a slower process in order to learn and select for the appropriate cells that are very specific to the problem at hand. So after talking about the innate response with neutrophils and the monocytes, and then the antigen presenting capability, we'll now focus on the T cell response, which is a really important arm of the acquired immunity. And you will see when we get to the actual therapies as a prime target for trying to optimize or regain an immune response to the cancer cells. Along those lines, we will again focus on and go into a little bit more detail on the antigen presenting cells, as well as the MHC molecules, those major histocompatibility complex 1 and 2, class 1 and 2, also known as human leukocyte antigens. So T cells start in the bone marrow, as we talked with lymphoblasts, and then they differentiate to immature precursor T cells. They get out into the bloodstream, and then through specific homing process, will find the thymus gland. And in the thymus gland, they get uptaked into the thymus gland, and they go through a process of selection, where we'll go through that in a little bit more detail on other slides. But as an overview, through binding to MHC class 1 or 2 molecules, they will be either selected because they can bind to them, but they will not be selected if they bind too strongly to them. And that is a mechanism that we have to protect ourselves to exclude T cells that bind to ourself too much to avoid autoimmunity. but at the same time, they have to bind a little bit to MHC1 or 2 because that's how our system surveils our body in order to eradicate external or internal problems. So they can't be too strong, but they can't be not binding at all. And we'll go through that in a little bit more detail. Now, the way that this works is that there's a specific antigen presenting cell in the thymus called a thymic epithelial cell. And this cell will help to select for T cells that have these properties. One thing that I'm going to show you is that these T cells that are immature precursor T cells, through a really fascinating mechanism, there are millions of possible receptors. And once a T cell is expressing one specific receptor, that's the only receptor that it will have, but that we have millions of different T cells that will each be expressing its own unique T cell receptor. And only a subset of those will get through the selection process in order to have these properties. And then the other part that we're going to talk about is they co-express a molecule, either CD4, which would make it a T helper cell, or CD8, which would make it a cytotoxic T cell. And we'll go into that in a little bit more detail. So how does the T cell become so unique, and how can we make more than 1 million versions of a T cell receptor? So this is a cartoon of what a T cell receptor looks like at a membrane of the cell. This is the membrane, this is the outside of the cell, and this is the inside of the cell. And the different proteins that come together, I mentioned how different proteins can bind like a lock and key, and they come from different genes, but they fit together, and they are noted here with different genes: TCR alpha, TCR beta, TCR gamma, TCR delta, and TCR epsilon. And each of those genes reside on different chromosomes, and they go through a process of transcription that is really interesting in how we get such variability in the T cell receptor. So as an example, the TCR beta gene, which again is on chromosome 7, is a sequence of a whole bunch of exons, they're called, on the DNA. And through a process of rearrangement of the DNA, different parts of this gene are selected and other parts are spliced out and removed or deleted over time. And so, as an example, this region is kept through the whole process, as is this yellow piece. But then different choices of this J region can be used, and only one of them is ultimately selected, etc. And so this then is the new gene after excluding all the other components. And every T cell will randomly do this in a different way, so that it will have a different variable region in the final gene product. And then that gene is transcribed, as we've learned, to messenger RNA. And then that messenger RNA is translated into protein, and then it gets into the receptor and binds to the other proteins that did the exact same thing in the other locus of the different genes. So all these different genes are going through this rearrangement randomly. And so there's so many different possible final outcomes of what this T cell receptor can look like. And so this is really important, because the variable region, which is usually this outer portion, as opposed to the inner portions, which are pretty constant, is that this will bind uniquely to a specific antigen in a specific epitope of a peptide or another antigen, because it is unique to that T cell receptor. Okay, so just as a quick summary, these T cells go through all these changes. They form a unique T cell receptor subunit, beta in this case, versus alpha, versus gamma, etc. And each of those are translated into protein that then come together and bind to each other to have a very unique epitope that they will recognize. And this happens in every single T cell, and that leads to millions of different versions of a final T cell with what it can recognize. The next part is, as I mentioned, a co-expressed protein here, whether it's CD4 or CD8, will determine if it's a T helper cell or a cytotoxic T cell. And we'll go into what the two different functions of those T cells are, because ultimately, when we talk about therapies are specific to usually these cytotoxic lymphocytes, which are the main effector component of our T cell response to getting rid of cancer cells. So we again looked at this now, T cells coming from the bone marrow, these precursors get into the thymus. They are going through this selection process by using antigen presenting cells, specifically a thymus epithelial cell. And so what exactly is that? And let's look at that in a little bit more detail. So an antigen presenting cell, again, remember, is a cell that can digest and repackage antigens or small pieces of protein called peptides. And they can do that either from an exogenous pathway, so little proteins or peptides that are floating around outside the cell that can either come from an external pathogen like a bacteria, or from a cancer cell that's ruptured and antigens are released. It can uptake this through endocytosis or phagocytosis and then process that protein and then express it on a molecule, MHC Class 2, and express it on the surface. And this will help to recruit T helper cells, those CD4 positive cells. On the flip side, all of our cells have the capability of expressing internal peptides called the endogenous pathway. And so all of our normal cells are taking degradation products of all of our normal proteins, expressing them on MHC Class 1 to our T cells. And if our cell is either infected by a virus, viruses get into our cell and live inside our cells. And so those viral proteins, as they're making them, can get digested and expressed on the surface. Similarly, cancer cells, which have become abnormal and have neoantigens, neoepitopes that I just mentioned in the previous slide, will also get digested and expressed on the surface. And so this mechanism is a way antigen presenting cells present through MHC Class 1. So all cells do MHC Class 1, and specific, or what are called professional antigen presenting cells, take external components inwards and express them on MHC Class 2. Okay, so different types of antigen presenting cells that we've already come across are macrophages and dendritic cells. We're going to get to B cells. And now this came up again in the thymus, for this thymus epithelial cell, and how it helps with selecting T cell during T cell development. So the thymus, remember, it's just in the chest area. It looks like this, and again, the T cells are coming from the bone marrow from lymphoid progenitors. They get into the outer component of the thyroid called the thyroid cortex, and through a process of selection, from the outer layer to the inner layer, these cells are either excluded because they have too weak of a signal, as I mentioned. They're not binding to either of our MHC molecules strongly enough. So if they can't bind there, then they're not going to function as they need to. They need to be able to function and surveil tissues. If they can't bind, then they're excluded. And then they select for either expression of CD8 or CD4. So they sort of differentiate and become either a cytotoxic effector cell or a helper cell. And then they go further down and go through another layer of selection, which is it excludes those cells that are binding too strongly. As I mentioned, this is a way to avoid autoimmunity. So we've excluded ones that are too weak, because that's how the process works. They have to be able to bind to the receptors, but it excludes also those that are binding too strongly to avoid autoimmunity. So the ones that are binding weakly or intermediately are the ones that are ultimately selected for. And so these final products, we're really going to focus on the effector products, because these are the ones that are mounting the immune response and ultimately often targets for immunotherapies. These cells then get out into the bloodstream. They're referred to as naive T cells. They've been selected. They will each be able to identify a unique peptide that they've been selected for, and they will go out into the periphery and surveil. And they will come across antigen presenting cells, either all of our normal cells through MHC Class 1 or MHC Class 2 through professional antigen presenting cells. And so a little bit more then about how this interaction with MHC class 1 and 2 is important to understand. So the next few slides are focusing on the MHC component of the immune system. So this major histocompatibility complex is a really important concept to understand because this is the central hub of how the immune system works. So the first is the class 1 molecule. So the class 1 molecule, as I've been mentioning, is expressed on all of our cells, and it is a way for our immune system to surveil our body and say, yes, this is our self, I will leave you alone. And so our cells will express this class 1 molecule, which is made up of two different proteins from two different genes, one on chromosome 6, where this MHC locus is, and this smaller protein in the class 1 molecule called beta-2 microglobulin on chromosome 15. This will become important when we get to the next video, which talks about mechanisms that cancer cells use to avoid our immune system. And so this molecule then is expressed at the surface, and this is where peptides will bind to in this this three-dimensional cleft that is formed by this protein. And so the function of it, again, is that our internal cells, our normal proteins from our normal cells, will get degraded, upregulated into this endoplasmic reticulum where the MHC class 1 molecule resides. And if your molecule is able to bind this antigen or peptide, then it will go out onto the surface and it'll express it there. Our T cells, which we've now selected from our thymus, will be patrolling. And remember, this peptide is being expressed inside the cleft of the MHC class 1 molecule, and it's very specific to the amino acids around this cleft. And so it binds like a lock and key there. And similarly, the T cell receptor has a cleft, and it will bind to this like a lock and key if it's the right lock to the key. And so if it is our self protein and it's normal, this will not activate, because remember, in our thymus, we excluded cells that bound too strongly to MHC class 1. And so this will not activate. On the other hand, if it's a viral protein or a cancer protein that is now being degraded and put onto the MHC class 1 and presented outside the cell, then if you happen to have a T cell that has a TCR variable region that can bind to this like a lock and key, then it will be activated. And it will say, this is an abnormal cell, it's got abnormal proteins, and I'm going to destroy it. So this system of monitoring our cells to exclude abnormalities is called cellular immunity. It's looking inside the cell and it's patrolling abnormal proteins that are there. And if they are present and you have T cells that recognize that as present, then the cell will activate. The other class 2 MHC molecule is also two different genes on chromosome 6 that come together as two components, an alpha and a beta component, and together they form a specific peptide binding cleft. And as opposed to the class 1 length of amino acids, which is usually around 7 or so amino acids in terms of the peptide length, this cleft will account for or accommodate a peptide length of around 13 to 18 amino acids. And so again, remember, this pathway of class 2 looks at the outside of cells and internalizes those components, digests them. And if you happen to have MHC class 2 protein that is able to bind this various antigens, it will then express them on the receptor. And this will then present to specific T cells, those T helper cells, CD4 cells. And so this interaction will then activate helper cells, which we'll get to as to why they can be useful and important in the immune response. So a little bit more about the MHC complex. So this MHC is also known as the human leukocyte antigen. And this is on chromosome 6 in the short arm of the chromosome. And remember, we have two copies of chromosome 6, one from mom and one from dad. So you have two regions. And on one region, one of the copies, you'll see that there's where the class 1 genes are, and then the MHC class 2 genes. And you can see that there are different subsets of class 1. There's locus A, there's locus B, there's locus C. This is a little bit simplistic. There's actually others. And then there's different loci for the class 2, shown here. And in contrast to how we get such diversity of the T cell receptor, where there's rearrangement of the gene and exclusion of all the extra components and selection amongst a lot of different choices to get a whole bunch of different T cell receptors, the way that the MHC becomes variable is that in the human population, there are thousands of different copies of each of these genes, which are called alleles or versions of the gene. And they vary at that region of where the antigen actually binds on the MHC or 1 or 2 molecule. So remember also that you have one copy of these genes from mom and one copy of these genes from dad. And so the idea is that, as shown here, you will have different versions of MHC class 1 from mom, different versions of MHC class 1 from dad, and similarly for class 2. And the idea here is that each of these different clefts will be able to bind to different peptides, different epitopes, different chunks of protein from all the different cells. And so that we're able to have a broad representation of as many peptides and proteins as possible. And remember that it's all functioning on this variable region of the MHC1. This is the version of MHC1, but the same concept for 2 is that it all depends on what this three-dimensional cleft looks like, which is based on the amino acids, which is based on the different versions that are available in the human population, thousands of them. And so this heterogeneity of the different genes and copies and versions is what is allowing us to be able to present a broad immune peptidome, it's referred to. So we can present as many peptides as possible on our receptors. And so I'm going to show you why that's important in a second. But again, a little bit more about haplotype. Haplotype is referring to what your versions are of each of these different loci. So that's your haplotype of locus A, B, C, D, R, D, Q, D, P. And you have 2 haplotypes, one from mom and one from dad. And this is just to show you the complexity of both the class 1 and class 2 is that there's thousands of haplotypes for the locus A, B, C, E, F, and G of class 1, and similarly for the class 2 loci. And again, this ensures a broad ability to express as many peptides as possible on your cells. And this study looked at what are the most common versions of each of these loci. And so they were comparing Caucasians, Afro-Brazilian, and Asians in this study. And they listed the top 10 top several versions of each of the loci. This is loci A. And you can see that the most common one was in Caucasians was about 27% of the population expressed this loci type 2. And it was a little bit different in terms of its percentage in the other ethnicities. And then similarly for locus B, the most common one was this 35 version, which was almost 14% of Caucasians, but much fewer Asians, for example, showing the variability across populations. And then in the DRB1 locus of the MHC class 2, the most common one being copy 11, about almost 15% of the population. And then what we're really most interested in is what's the most frequent haplotype. So the combination of all of these different versions of these as one. And you can see that even though, for example, locus A is 27%, locus B is almost 14%, this DRB1 11 is almost 15%, to have all 3 of them in the same haplotype, on the same chromosome from the same parent is only just over 1% of the population has the most common. So what I'm trying to get at is there's extreme variability from one person to the next in terms of their haplotypes, in terms of their immune system, MHC class 1 and 2 molecules. And so any one individual is very different from the next person. They'll be similar if you're in the same family, but more likely than not much different from each other. The only people that would have an identical immune system are, of course, identical twins. And the most important part why I'm going through all of this is this statement here is that this can explain why For example, some people had extreme illnesses to COVID-19. They have, that, for example, may have an immune system that isn't able to express the COVID-19 proteins as well on the surface of their cells. And so they can't mount as good of a response as opposed to someone who didn't have much of a problem with COVID-19. They had immune systems that were already able to deal with this problem, express the proteins properly, and have a nice response of their immune system to the COVID-19 and clear it. Similarly, and relevant to what we're talking about, this may explain why people are at higher risk of getting cancer. For example, the same cancer, the same gene mutations may have a different response in a person that has a given immune system compared to another. If they can't express the cancer's proteins on the receptors, our immune system can't recognize them as abnormal and can't mount a response to the cancer. And so different people with different immune backgrounds can be more susceptible or not to different cancers. And lastly, and importantly, this is also certain haplotypes of these immune MHC molecules have been linked to having a higher risk of autoimmunity for similar reasons. They're too strong of a response to their own cells, and they're not able to exclude those cells in the thymus and other areas to be able to dampen a response of our immune system to our own cells. This interplay between cancer and autoimmunity becomes very important when we get to actual therapies, because when we're trying to treat cancer by stimulating our immune system, we can then have the negative consequence of stimulating it too much and having autoimmunity, where the immune system is now attacking and killing our own normal cells. And we'll get into that into later videos. So in summary, because T cells are so important, we spent a lot of time here, but what you should take away is that T cells start in precursors from the bone marrow. They home to the thymus, that gland in the chest. In the thymus, they are selected so that they are binding a little bit to MHC molecules 1 and 2, but not too strongly, because we want to avoid autoimmunity. And that there are millions of these precursor T cells that occurs through the gene rearrangement that we went through. So they each express a unique T cell receptor, but only a subset of those get through the selection process. But when they do, these naive T cells that have not been exposed to an antigen now in the real life setting, but they're going to go out into the bloodstream and monitor our body through the class 1 and class 2 mechanisms to ensure that it removes abnormalities that are either from outside, from pathogens, foreign antigens, or from external problems that it can also be from a viral infection or cancer. And we talked about the receptor on the T cell that is a CD8 positive, or a cytotoxic T cell that has CD8, and that will bind to a specific peptide that is expressed by MHC Class 1, which we went through, that would be expressed on an infected or a mutated cancer cell. And we went through the MHC Class 2 axis, which binds to the T cell receptor on CD4 helper cells, again with specific peptides, but through this professional antigen presentation that these helper cells, as we'll see, can then recruit other T cells and B cells, as we'll get to. So again, these cells get out into the bloodstream and they start to patrol. And remember, they're going to come around and patrol cells. And if they, for example, come into contact with an antigen presenting cell that has a cancer peptide that it's taken up in the lymph node, and it's expressing that abnormal protein, if your T cell and T cell receptor happen to recognize that, then this T cell will be activated and proliferate, and then it will go out of the lymph node through the bloodstream and start patrolling the body to find that cancer that's expressing this very antigen. When it gets there, this cytotoxic T cell that has this TCR that's binding this specific antigen, if it finds that cancer cell or infected cell that has expressing this antigen through its MHC1, that is a prerequisite interaction in order for this to get activated. We also have to take into account a whole host of other protein-protein interactions through receptors on both of the cells that I can either be activating or can be inhibitory. And ultimately, it is the balance of these activating versus inhibitory signals that will then tip in favor of proliferating and activating and killing versus shutting this interaction down if there's an overwhelming amount of inhibiting molecules. And again, these molecules will become important when we get into how cancers evade the immune system, and in the third video, how we can overcome that with targeted therapies. These extra pathways, these extra binding of proteins are called checkpoints. And so these checkpoints are all evolved to really be sure that we really do want to kill this cell and to avoid autoimmunity. So many of our cells will co-express these receptors to protect themselves from our own immune system attacking them. And we will learn that cancer cells hijack this to be able to, one of many ways to evade our immune system. So to summarize what we've talked about so far in this cancer immunity cycle, we talked about cancer cells in the tumor, how they're proliferating, they're dividing, there's some cell death, there's some release of cancer cell antigens, DAMPs, and this will travel through the lymph system to regional lymph nodes. These lymph nodes have a lot of different immune cells. One key cell there is called a dendritic cell, which is a professional antigen presenting cell, will suck those proteins up and express them on their receptors in MHC class 1 or 2 molecules. And if it's an MHC1 molecule, then it will recruit and bind to cytotoxic T cells, and this will select for that specific T cell receptor that recognizes this antigen and create an army of those T cells that then get back out into the bloodstream. They have to be able to traffic and find the cancer cells, get into the area where the cancer cell is and infiltrate there, and then recognize the cancer cell through that specific binding of the T cell receptor and the MHC class 1 molecule with the antigen in the pocket, and have co-expressors conducive to a T cell kill. So this is what is supposed to happen as it goes through this cycle. When we have a cancer, our immune system should recognize each of these steps and kill or eradicate the cancer. In the upcoming video, we're going to learn at each of these different steps how cancers can evolve to evade our immune response. So in this very complex talk about the immune system, we first learned about the precursor cells of the immune system and other blood components. We learned about the innate immunity, which is that first response with neutrophils, macrophages, and that will help to temporize the problem, but also that many of these cells, like macrophages and dendritic cells, can uptake these peptides and start to present them to select for specific T cells. And now we're going to learn about B cells to elicit an immune response that's more adaptive and specific to the problem at hand. One thing to mention is that after, say, an infection is cleared or a cancer is cleared, with all of those T cells that we've selected for that were specific for that particular problem, these cells dissipate and go away, but there will be a small subset called memory T cells that linger in the event that we will have a re-exposure to that same infection or to another cancer like that, to try and spark that response in a quicker manner. So now we're going to talk about one more component of the innate immune response called natural killers. And remember, these are cells that are actually from lymphoid line, but they are involved in what's considered innate immunity. So in contrast to the T cells, which the T cell receptor and the MHC molecule is a activating response when it binds, for the NK cell, they actually have an inhibitory receptor that binds to MHC molecule 1. And so when this binds, the cell will not kill a normal cell. Why this is important is because when our normal cells are infected, say, by a virus, and the virus is residing inside of the cell, or you have a cancer cell that has abnormal proteins, mechanisms are in place for it to downregulate MHC molecule 1 expression. And so this is one mechanism for it to evade the T cells. T cells can't see them. They're not expressing MHC1, and so a T cell is not able to see it. But we have a backup plan where NK cells if their inhibitory receptor is not recognizing the cell, it's saying there's no MHC1 molecule. This is not us. This is not me. It will then go and kill that cell. And this is one of the many ways that you can have graft rejection in a transplant, say of a liver transplant, where you have someone else's liver cells there that are expressing different molecules, obviously, of MHC1 because they have a different immune system, and your NK cell doesn't recognize it, and so it says it's abnormal, I'm going to kill it. So you need to give suppressive medications to suppress this response in that situation. Now, this is a complex interaction, and what we see here is quite simplistic. In fact, there's many different receptors, as we saw with T cells, that are either activating or inactivating, and it's the balance of these that will then tell the NK cell to proceed with killing or not. And in fact, this is a simplistic look, whereas we really see that there's numerous activating receptors on NK cells and numerous inhibitory receptors, and it's, again, the balance of whether there's more of one or the other that ultimately would determine whether the NK cell will proceed to kill the target. And don't forget, we also talked about, when we were in the targeted therapy section, when we talked about using targeted antibodies, and that one of the mechanisms in which antibodies work, say trastuzumab to HER2, is that they recruit natural killer cells, NK cells, through the back end of the antibody, the Fc domain of the antibody that binds to the Fc receptor, or the CD16, also known as. And so this will then activate the NK cell through antibody-dependent cell-mediated cytotoxicity. So they can act through various receptors directly on the target, and or they can be recruited to the target by antibodies. So the last type of cell that we need to understand is the B cells. And remember, they start from the lymphoid, they go through B cell progenitors, and then, as I'll show you, when activated, they will become plasma cells and make antibodies. And these B cells, very much like the T cells, have a receptor instead of a T cell receptor. It's called a B cell receptor. And the B cell receptor is actually the antibody that's expressed on the surface of the B cell. And the antibody is composed of two proteins. One is called the heavy chain, and one is called the light chain. And each of the genes is on different chromosomes. And very similar to T cells, they become very variable based on this rearrangement of the gene that leads to a variable heavy chain and a variable light chain. And when they come together, they form a very variable region that is where they bind to antigens, and then a constant region. And so again, this is important because it leads to such profound diversity of the antibodies and what they can recognize. And so, similar to T cells, they start in the bone marrow, and much of their selection, as opposed to T cells in the thymus, is in the bone marrow, and some of which occurs later in the periphery as it's floating around in the bloodstream. And so, similar to T cells, there's a selection process. Once the B cell has committed and it's spliced out all the extra DNA and it's committed to just what it's going to be transcribing and then translating into protein, it will then go through a selection process. And again, we want to exclude those B cells that have a B cell receptor antibody that will bind too strongly to our own cells, our own antigens. And so those cells die through energy. They're not selected. They don't need a positive selection, like they don't have to be able to bind to MHC1 or 2 like T cells. So most of it is just negative selection. They're getting rid of these ones that are too reactive. And then the remainder of them get out into the periphery. And as I mentioned, the antibody sits as a receptor in the cell membrane. Again, because of this rearrangement that occurs in both of these genes, billions of combinations are possible. And so antibodies have such a wide portfolio of antigens and epitopes that they can bind to. And again, a subset of those are excluded because they're reacting to ourselves. But then we have all of these B cells that come out that are considered naive B cells. They've not been exposed now to real life antigen, but they're prepared. And we have a whole portfolio of them that we can choose from in the event that we need them. And this is summarized here where we start with the stem cell that goes to precursors, and then through that gene rearrangement, we'll create unique B cells that recognize a unique antigen on the B cell receptor antibody. And we have literally billions of these versions. To think of billions of different colors, and each one is specific to a given antigen or epitope. And then these naive B cells that are floating around in the bloodstream, if they come in contact with an antigen that they happen to recognize, they're selected for and activated. So upon activation, these cells can then undergo proliferation so that you create an army of this B cell that has the same B cell receptor that was selected for by the antigen. This is facilitated and accentuated by helper T cells that bind to, through the MHC class 2 molecule, binding this lock and key with this peptide fragment. And then this army of B cells that have been selected for will then differentiate and become plasma cells, which then go back to the bone marrow, set up shop, and secrete antibodies of this specific peptide. And so they can go around and through our body and neutralize the problem that wherever it comes in to counter them. This system of the immune system is referred to as humoral immunity. So the B cells are creating antibodies that get out into the bloodstream, and each plasma cell can create numerous amounts of antibody. In addition to that, these B cells can secrete cytokines, and we're going to get to that in a second. And these cytokines can then recruit other immune cells, other T cells, other macrophages. And so there's this positive feedback. Once a cell is activated, it will then secrete cytokines that then recruit more immune cells to the area. And then finally, akin to how T cells have a residual memory cell after the problem goes away, in case it encounters this problem again, it will be able to mount the response quicker. And that's the same thing with these B cells, is that this process takes time to select for the B cell, to proliferate it, and then to differentiate and then create antibodies. If you've already got a memory B cell that already has that specific antibody at B cell receptor that binds to and recognizes a specific antigen, if you ever come encounter with it again, then this will proliferate much more rapidly. And this is, again, how vaccines work. Many of our vaccines create these memory T cells, and so that they're ready in the event that we do get exposed to that antigen in the future, we can mount a response a lot quicker than over a period of weeks, like what takes for the adaptive immunity to normal take place. So the last topic to talk about is cytokines. And I mentioned that briefly on the previous slide. And that what you see here is a very complex interaction of all of the different types of cells that we've talked about. And it all starts with the central problem, whether it's a bacterial infection or viral infection or cancer, where there's an abnormality that's recognized by one or more of these different cells. And when that happens, you see this positive feedback loop of all of these cells talking to each other by secreting factors called cytokines. Many of the common ones listed here, like interleukins, interferons, tumor necrosis factor, etc. And these all rev up our immune cells and help activate them and home to wherever the problem is. This is referred to as inflammation. With all of these cells being upregulated, it is a defensive response by the immune system, dispatching all of these various different types of white blood cells to the affected areas. And from the patient's perspective, they can, and say in the setting of an infection, they can experience redness and swelling in that area and fever. And these symptoms are a consequence of these cytokines, which can often cause fever, for example, and can cause the redness and swelling of by those cells infiltrating into where the infection is. We should also keep in mind that although there's a lot of positive or activating responses and cells, there's also the counterpart regulatory component. We've talked about that when we saw the T cells, how there's checkpoints that will keep it in balance. NK cells have checkpoints to keep it in balance. And we also have cells that are regulatory T cells, regulatory B cells that keep this whole process in balance. And when there's a appropriate response to be had to an infection or cancer, it will tip in favor of an activating response. And similarly, if we don't want to have an activating response, then it will be turned off. And we get problems in cancer when this is dysregulated inappropriately. So with this understanding now of the complexity of the innate and acquired immune system, we picture our cancer starting in the stomach or the esophagus and draining to regional lymph nodes, the different peptides and the different damps from the cancer cells. And in that lymph node, we see special cells of the immune system, specifically dendritic cells, that are taking those up and teaching or selecting for our army of T cells that will recognize those antigens and select for them and activate them through these MHC molecules that are specific to the T cell receptors. And this will help to recruit all of those cells that we just went through to effect a response and eliminate the cancer cell. This is the cancer immunity cycle of what is supposed to happen. And hopefully we can recognize the complexity of this system and how fascinating it is to be able to have such a diversity in response, to be able to accommodate and recognize various problems that we might encounter to be able to neutralize them. And so this cancer immunity cycle, we see all of the steps that have to happen in order to have an appropriate immune response to the cancer. And now that we understand that, in the next video, we're going to be looking at the various ways that the immune system is evaded by the cancer cell through each of these different steps. And in the last video, what are some of the strategies that we have from an immunotherapy standpoint to regain control, to tip the favor and balance of an immune response to the cancer. So in this video, we went through the complexity of the immune system as simple as I could. And believe it or not, it's even more complex than what I've presented. But this is a general framework of understanding that you may need to watch this once or twice to get the full meaning. And so we went through the innate system, that is the first line of defense, but also starts to get cells like dendritic cells that start to teach or select for the acquired or adaptive immunity that's much more specific and targeted to the in our case, the cancer cell, using MHC molecules and the cytokines that these cells all talk to each other in order to mount an immune response to the cancer. And that this response is a balance of activating versus inhibitory signals on the immune cells. And ultimately, when we get to immunotherapy section, we're going to see how we can balance that in favor of an activating response to the cancer. So in this video, Principles of Targeted Therapies Part 1, we went through the basics of the immune system that are required to understand what an appropriate immune response to cancer should be and how it's done. And then in the next video, we're going to look at the various ways that a cancer can evade our immune system, and following that, a video on various strategies to be able to harness the immune system to regain control and to recognize the cancer as being abnormal and to eradicate it. So stay tuned for that. Thank you.